Hi, my name is Kristin, and I have a confession to make. I am that thing that we're all trying to escape and get away from because of the wonderful new technologies we have and the resolution we can achieve. I am the fruit smoothie. But I've decided I'm going to embrace my inner smoothie because the project that I represent as this smoothie has a lot to teach us. It was a forerunner to the Human Cell Atlas that we're all here for, and there's a lot of lessons to be learned, a lot we can leverage from it, and a lot of things we can take from it and actually apply here. So I'm going to talk about that project, which is the Genotype-Tissue Expression Project, and the goal of that project was to understand the regulation of gene expression, specifically to characterize the regulatory architecture of the human genome, and to ask what role genetic variation across this human axis played in regulating gene expression across this sort of multi-tissue axis in a range of non-diseased human tissues. So we wanted a reference normal. So the challenge in doing that, to create that reference map, which has parallels to what we're discussing here, is that we needed to be both multi-tissue and multi-individual, and that set us in a place where we could only achieve this by surveying deceased donors. To collect all of these tissues from many individuals, we had to survey deceased donors. So this is the sampling design and project design of the project. We needed to enroll donors, so we enrolled 960, actually a total of 980, I believe, recently deceased donors, and created an atlas of gene expression and gene regulation. We sampled up to 53 tissue sites, more commonly in the range of 30 to 44 per individual, which included 11 distinct brain subregions and 2 cell lines as well. And the core molecular analyses we did were a whole genome so that we could get that genetic variation and survey that across individuals, and RNA sequencing of all the tissues, the bloods, the cell lines. And more recently, we're just producing small RNA and microRNA sequencing across all of these tissues as well. In addition, and I won't talk about these, there are a suite of additional secondary molecular analyses that were largely epigenetic and protein-based as part of the enhanced GTEx project. And I will note that all of the methylation assays were actually done on sorted nuclear cell populations. So this is the sample collection pipeline. Aviv referred to GTEx yesterday, but I think she actually mistakenly referred to GTEx as only collecting these sort of post-mortem donors on that axis from fresh to totally post-mortem. GTEx collected basically anyone who was deceased across the spectrum. So that included both organ and transplant donors as well as the rapid autopsy post-mortem. So these latter tend to be more accident and trauma victims. They're rarely, only occasionally vented. Collection teams were assembled immediately. They tend to have slightly longer ischemic times for the tissues you collect. At this end, the transplant donors were those who had significant brain damage or tended to be brain dead. All of them were vented, and so at the time of cross-clamp, they were perfused so you could preserve the organs. And we had collection teams ready in the operating room, ready to go in after those, so they had much shorter ischemic times. We obtained donor consent through next of kin donation for the study. And in this particular instance, because this was the first time anyone was sampling this many organs from this many individuals, we actually obtained consent where we said we would keep the data in a protected database. However, as part of the project, we did trial a small subset of donors with open consent and showed that we successfully could do that. We collected 4 donors with totally open consent for this project. We surveyed across racial and ethnic groups and sex and age range and collected brains that were not on ventilators. So this is what the operations looked like. As I said, we had a team. It was highly organized when we went in after a donor, so we could get this many samples of high quality in a very short time period. They were normal, non-disease reference population was the goal. And really what we needed to do was rigorous, repeatable sampling so we could get an accurate reference map across Okay. So, as I said, we needed to, one of the same challenges here in the discussions we've had with a common coordinate framework is GTEx needed to address that as well. We needed to be able to sample these sites repeatedly from donor to donor. And this is an example from the GTEx portal for one of the sites we sampled. Here is the small intestine terminal ileum, and you can see that the sampling site is defined as the thickened mucosa corresponding to lymphoid nodes, the Peyer's patch, just proximal to the ileocecal valve in the most distal part of the small intestine. So we had to come up with a set of instructions and protocols that people could follow to try to survey the same. And that's delineated here. We had developed extensive SOPs and tissue harvesting protocols. The team had to collect multiple tissues, different fixation and freezing types, some for histology. And so we developed an SOP that describes the precise site and size, where to sample, what to avoid, what size to get, and so on. And this is just a quick breeze through for other organs such as the heart, and the gastroesophageal junction and so on. And all of this is available, and all of this is leverageable as sort of a baseline going forward as we go deeper into these tissues that are being discussed here. So this is roughly the sample collection pipeline for any given donor. We collected several specimens that were fresh, bloods and fresh skin that were shipped immediately, as well as brains that were shipped immediately for subdissection. But I have an important note here that this could include other tissues. It wasn't in the case of GTEx, but this could easily be adopted to include other fresh tissues. And there were many other tissues we collected that we then preserved and had to bank for the project and for the analyses. I want to just discuss most of these, which are the bulk of the tissues. One of the important things we did was pathology review of every tissue. This is one of the largest databases of a pathology reference of normal tissues that there is, because normally you don't get this sort of histology and pathology review on this many normal tissues. So we did this of every single tissue, and there was a matched tissue piece for each sampled tissue. So I'll just spend a few moments on that because that was an incredibly valuable piece of data for GTEx. So here's a tissue being collected. Here's the size demarcations and the cassettes that it's going into. So very specific cutting and size of the tissues. That was illustrated here. At least one of those sections was made into a slide, digitized. We had pathology review performed on all of those, and all of those are actually available, and there's a lot of people using them for image analysis right now. So this is the sorts of things we did in the pathology review. They would confirm tissue type, purity, the correct tissue and sampling site location, or comment if it wasn't, sample size and number, the autolysis and preservation level of the tissue, any lesions and disease would be commented on as well. This is from the SOP, which is basically during procurement, GTEx aliquots should be taken from tissue that appears grossly normal because we wanted a normal reference. However, even if no normal tissue is apparent, the organ should still be sampled and abnormalities documented on the CRF. So that sort of variation we discussed before in the lung, the sampling protocol in GTEx was to sample all of that, but to document it as well as possible. And we collected older individuals, and many of those, as you might imagine, had chronic lesions that include congestion in the lung, scars from MI, smoker's lungs, renal scarring, muscle atrophy, and so on. So the general rule for inclusion in GTEx, which pathologists perform, was a tissue was acceptable if it had these chronic lesions without extensive tissue loss, but that was documented, and it was unacceptable if we encountered diseases with extensive loss of normal tissue or advanced cirrhosis, neoplasms, and so on. That said, we didn't discard those tissues, we kept them, and I'm sequencing them regardless. So another thing that pathology did was to assess and estimate the cellular heterogeneity. Here's an example of one of our stomach samples. As you can see, the mucosal glands were the goal that we were trying to sample, but in this piece, as in many, we often also sampled excessive muscularis as well. And that's the type of thing where there was good correlation with cell types evident in the RNA-seq data. We went back and did some laser capture to show that these are all largely predominantly mucosal cell populations we were sampling, and these were largely muscularis. So those drove the first couple of expression principle components in the lung. But even with these rigorous SOPs in place to collect the same tissue from time to time, it gets back to Aviv's point yesterday that in a short person compared to a tall person, sampling the same piece of tissue in roughly the same place may still give you differences between individuals. And that's actually illustrated here. We can see that in the GTEx database. If we look at 3 tissues, breast, heart, and lung, here we have an example with the pathology comments and the views. This is mainly fibrous with ducts and lobules, whereas this one is predominantly fatty and only 10 to 20% of duct lobular elements. Here in the heart, we have 2 pieces, no abnormalities, mild chronic ischemic changes and extensive scarring, and likewise across the lung. So despite these sampling in the same locations, there are still extensive differences. However, that said, those differences may still not drive most of the expression differences in the comparison between these. And one of the things we've been discussing with one of the DCP groups, and I can't remember which color it was, is that capturing the metadata surrounding these is also very important. GTEx captured an extensive amount of metadata in addition to all this pathology and diagnostic thing, and that proves to be very important in sort of disentangling and comparing these. And this is just an example, again, of those 3 tissues that you saw the variation in: breast, heart, and lung. And what's driving the major expression principle components in breast is obviously sex is the key one there. That's not the case in heart, where warm ischemic time is one of the main drivers of the first and correlates the first PCs. And in lung, it's the cause of death, which is delineated by the Hardy scale here. And you can see that whether they're on ventilators or not was a major driver there. So I'll just finish up. This is one of the things, having put this network into place, we can leverage it for GTEx, and it's one of the things we're hoping to try to do, not for GTEx, for the Human Cell Atlas, to go back and reuse some of these and adopt them for some of the collections that we can do in the Human Cell Atlas. But there are additional things we can leverage from GTEx, and I just have a few slides to that. And one of them is the extensive data we have on that axis of how representative you are of a living tissue when you go from transplant to a postmortem organ donor. We have extensive data about ischemic times across many of the tissues, because in GTEx we collected them across all these times, depending on what we could get. So that data is available. We can leverage it. The analyses we've done has shown that the expression of most of these genes was not significantly correlated with the ischemic time, and that the genes affected tend to be tissue-specific. But there's a very large amount of data here we can go back and look at within each of these tissue systems. One of the things that was done is a comparison of GTEx with the Human Protein Atlas expression data. All of the data in that atlas, which sampled many of the same tissues, was collected from independent surgical samples. And this analysis was done by Matthias Uhlen, and you can see that there was extremely good and high concordance and repeatability between the expression of the same tissues sampled in the Human Protein Atlas and in GTEx, suggesting that the recapitulation of living donors was very high in the deceased postmortem tissues we collected, and also that there was good repeatability and correlation between these differently sampled sites, the ones we sampled and the ones they sampled. And where we see differences both in the brain here and in the skin, they actually reflect sampling differences. In the skin, we sampled through multiple layers, whereas the Human Protein Atlas had a skin shave across just the top layer. GTEx also collected 5 matched tissues from a subset of living donors, so we have data on that. And we ran a biospecimen method study where we sampled 6 tissues over different time points. So there's a lot of rich data there we can leverage and to inform the sampling for this project. Another thing we can leverage is the knowledge of individual variation. Obviously, that's not a part of this first atlas for very good reasons that many of the speakers have addressed, but there's a lot of information on it. 2 projects, one is called the NTEx project, which was a project where we deliberately joined GTEx and ENCODE together by co-sampling 4 donors between the projects. So the goal there was to link those 2 projects and to link the functional maps to the individual level genetic variation. And what ENCODE's been doing is extremely deep genetic characterization of those individuals, and they show a considerable amount of genetic and structural variation at the individual level. And then GTEx itself, the whole goal of GTEx was to look across individuals, and the two take-home themes for that we published this week is that the common genetic variation influences gene expression of 90% of the known protein coding genes, and that rare variation also has a big effect on gene expression. So this is the type of information we can leverage as you go forward into more of that individual comparison and individual access. And in addition, we can also leverage the banked samples that are left in GTEx. And this is an example that I've been doing in collaboration with Arit and the group that's at the Broad with Aviv and Arit. And what we did, this was published just recently, is to take a set of GTEx samples where we had frozen tissues and we surveyed them from the hippocampus and the frontal cortex, and to take one of those beautiful new sort of nuclear dissociation methods that work on frozen tissues and to test it out. And as you can see here, this was published in the paper. We did several donors, and these were the same donors we had bulk sequencing on, and we have beautiful differentiation of the cell types in the hippocampus and prefrontal cortex, showing that we can go back to some of these banked frozen tissues, which was really nice. This is an ongoing pilot. We're now surveying all of the tissues that we actually ended up freezing in GTEx, and those include the heart, esophagus, lung, breast, muscle, skin, and prostate. And all of these are challenging in their own ways, but we're trying to see how these methods can apply to all of these different tissues. And then finally, one of the things that GTEx did that I think came out a lot, we had a series of papers come out last week. One of the things that GTEx did in enrolling donors is we actually engaged the donors in the study. And this came out in several articles. There was a Nature podcast and several reviews here with some of the donor families who donated loved ones to the project. And they turned out to be very, very open in describing the loved ones they donated, the reasons they donated, and what was important to them in the project. And as you can see, that many of these donor families attended the project's annual meeting last June. They met the scientists and they're engaged in the research. We engaged with them, and to this donor's wife, to know that he's still there is a wonderful thing, she said. So this is something that we found was very important in doing a big-scale project like this, that the donors' families and the people who are engaged in it are very willing to be part of these projects. And I think if you're asking these individuals to consent to open access, they're quite willing to do it, but they'd love to be engaged in it as well. So I'm going to end there and acknowledge the GTEx Consortium, which is very large, many of the people who made it happen, and all of the people who helped collect, and these are the people we've been talking to, and particularly the donors and their families. As I say, this was a huge contribution and an amazing resource that they made available. Thank you.